The following is a tutorial for the cellular localization module of IMG-ACT. So I'm logging in to my class here, and I have my assignment, just like all of you. I'm going to go to my first lab notebook link, and I'm going to move right on down to the cellular localization module. There's some information I need to fill in here in order to make this work. Just like before, I'm going to need to go and have my protein sequence handy, so I'm going to make sure I get that. So now I've copied my protein sequence. I'm going to go back to cellular localization. The first item that you are asked to get is the gram stain. The gram stain is something that we're not going to discuss in this class. It is a primary wet laboratory tool for the classification of bacteria. And the reason that we need to know the gram stain in this particular case is because we need to use that piece of information in some of these other bioinformatic tools. So next, I'm going to open up this link, which is a transmembrane search tool. So while this is running, in essence, what we're doing here is we're taking that protein sequence and we are examining the amino acids in their order and comparing the order of amino acids in this primary protein sequence with a vast database of other protein sequences that go across bacterial cell membranes. In this particular case, we see a result that is not initially intuitive, other than you can see that red would mean a transmembrane portion of the protein, blue would mean a cytosolic or inside the cell portion of the protein, and this represents something that's predicted to be outside of the protein, which is kind of odd. But let's just go back here and say number of predicted transmembrane helices. Here we have 0 or 0. The transmembrane topology graph. Okay, the number of expected transmembrane helixes, that's where I got that, 0. This topology graph, when we continue with our basically copy image and we pray that it will paste in here, which it has done. And I comment this protein or polypeptide has no apparent transmembrane domains, but it does oddly appear to be a protein that is outside the cell. I'll save that because, and you're doing the same protein, so you can type those same words in there. Here, again, there's a lot of information here that I'm not going to talk about too much in this situation. A reminder as we move forward to the signal protein peptide part of this, that there is a text tutorial up here in the module quick links. So let's move on. I'm going to close this tab now, and I'm going to open up the, whoops, I missed signal P. I'm going to open up this tab. Same kind of story here very quickly. Oh, I don't have my, once again I've lost my protein. So back up to basic information module. Need to get my protein. Cellular localization model signal server. We're going to paste the fast. And now here's where you need to remember It's gram-negative bacteria, or gram-negative bacteria, different kind of cell membrane, different kind of cell wall. This one's searching for a signal peptide, something to send the cell outside. And really what we see here in a crude thing is no. The answer is there is no evidence to predict this. Without going into more details about how this works, I want to keep this tutorial quick. The signal peptide probability value, the signal peptide probability value here is 0.144. Oh, so I'm going to put 0.144 in here, and of course then there's no cleavage site, and I'll just grab the graph by copying the image and pasting it in here and saving. Now I've successfully saved this particular part. Now we'll go on to, as always, I can continue to forget that I need to keep my protein sequence in my sort of copy and paste memory. And so we're just going to go get that, come down to the cellular localization module, open this in a new tab, and here we are again. It's a bacteria, it's a gram-negative bacteria. I'm going to copy and paste this guy right in here in the FASTA format, and I'm going to submit it. My email address doesn't seem to be valid. When did we start doing this? Oh, I don't want to send by email. I want to view it via the web. So it's not going to let me do that, so we'll just go back. I'm just going to quit that and open up a new one and say send via the web. I want to make sure I have a gram-negative. And I'm going to choose a bacteria. It's gram-negative. I'm going to paste this guy in here, and I'm going to submit. Okay, so here we have a lot of unknowns and no details. So this particular protein has a localization score here of probably being in the cytoplasmic membrane of a bland gram-negative bacteria. Let's go over and see what we get for p-sort. Cytoplasmic score, well, let's, sorry about that. Basically all these scores are 0 except for cytoplasmic membrane. So 0, 9.98, 0, 0, 0, 0, final p-sort prediction, cytoplasmic membrane of this gram-negative bacteria. Save. Cytoplasmic membrane. I might just pop this in there as well and close. Whoop, and close this. Paste it in there just as my lab notebook so I don't forget, because it's possible to forget things. Finally, I'm going to go up to Phobius, another tool. So let's keep in mind that what we have done is we are investigating the cellular localization predictions of a protein that's predicted to exist in a genome. And we have done this with 4 separate tools that are available here, combined transmembrane topology and signal peptide predictor. This particular output will combine both signal peptide and transmembrane output. This is sort of another way of doing it. So this is sort of in the idea of there's more than one way to solve a problem, and when there's more than one way to solve a problem, we try to make sure that we show you a couple of different different mechanisms. Wow, so here we have the Phobius graph, which I'm going to copy. I'm going to paste that graph right in here, and the prediction based upon these tools, based upon the work in this module, I predict that this protein is found in the cytoplasmic membrane. Save. Okay, now I've completed this cellular localization data for one of my assignments here in this lab notebook. I'm going to need to go to my lab report. I obviously am a little deficient here. We're going to have to get some information and plug that information there, but I'm confident that you guys can figure this out on your own.